Now nitrogenase enzyme, it has two subunits. One is the larger and another one is the smaller. अब जो larger subunit है इसके अंदर present है iron and molybdenum. So it is a iron molybdenum protein part, and the smaller subunit it only possess iron protein part. अब जो larger subunit है it is tetramer, yes, and the smaller subunit it is a dimer. Larger subunit के ऊपर present है active site for the attachment of nitrogen, and on the smaller subunit there is presence of the active site for the attachment of ATP. Yes, nitrogen fixation, it is highly energy consuming process. तो nitrogen fixation के लिए there is requirement of the energy which is provided by the ATP. So here I will be making the diagram of nitrogenase enzyme, and for the purpose of the simplicity I will be showing here only one site where there is attachment of nitrogen. This is nitrogenase enzyme. In the nitrogenase enzyme, this is binding site. Yes, this is binding site for the nitrogen. Now, under the anaerobic condition, this binding site, it is acquired by the nitrogen. So you know this very well that in the nitrogen, that is dinitrogen, the two nitrogen, they are joined with the help of the triple covalent bond. Yes. So there is presence of three covalent bonds between the two nitrogen. Now this nitrogen, it will undergo reduction. Reduction होगा, there will be addition of hydrogen ion, there will be addition of electrons. तो जब hydrogen ions electron add होंगे, so one of the covalent bond between the two nitrogen that will break. So after the reduction, so now what will happen? One of the covalent bond that will break, and there is addition of two hydrogen. तो दो में से एक कोवलेंट बॉन्ड ब्रेक कर जाएगा, and there will be addition of two hydrogen here, and this compound which is formed, that is N2H2, and this compound, this is known as diamide, right? Now again there will be reduction. Again reduction होगा, दो हाइड्रोजन आयन, दो इलेक्ट्रॉन्स, नाइट्रोजन एक्सेप्ट करेगा, yes. So when again reduction will occur, 2 more hydrogens, they are added. So what will happen? Again, one covalent bond that will break. So one more covalent bond that will break. So there is addition of the 2 more hydrogens. 2 more हाइड्रोजन ऐड होंगे और ऐड होने के बाद अब जो कंपाउंड बनेगा दैट इज N2NH4. N2H4, and this compound which is formed, this is known as hydrazine. Now, ये जो रिडक्शन का प्रोसेस तब तक चलता जाएगा till the time the nitrogen it is reduced to the ammonia. So now the last covalent bond between the dinitrogen that will break due to the reduction process. Yes. So now the last covalent bond that will break due to the addition of hydrogen. Yes, so there will be formation of the 2 molecules of ammonia after the reduction, 2 molecules of ammonia, and you have to keep this in mind. There is also formation of the one of the byproduct that is hydrogen. और इस byproduct को भी form करने के लिए there is requirement of additional 2 electrons and 2 hydrogen ions. So you're getting this point, that nitrogen, after accepting the electron and hydrogen ion, there will be formation of the 2 molecules of ammonia, and along with that this formation of a byproduct, and for the formation of the byproducts there is additional requirement of 2 electron and hydrogen ion. So let's write the complete equation for the nitrogen fixation. For the fixation of 1 nitrogen, there is requirement of 8 hydrogen ions. 2 plus 2 plus 2 and 2 here, and along with that 8 electrons, and for the transfer of 1 electron there is requirement of 2 ATPs. 2 ATPs are required, so you can say there is requirement of 16 ATPs. 1 electron के लिए 2 ATP, तो 8 electrons के लिए 16 ATPs they are required. Now nitrogen, after the reduction it is converted into the ammonia. The 2 molecules of ammonia they are formed. Along with that there is formation of the byproduct that is H2. अब ये जो ATP है ये कैसे एनर्जी प्रोवाइड करेगा? Now one of the high energy bond in the ATP that will break. One of the high energy bond in the ATP it will break, that is the one by which the phosphate is attached. So that bond will break and the ATP it is converted into the ADP plus PI. ADP, adenosine diphosphate, PI, that is inorganic phosphate. So there is formation of 16 ATP and 16 PI. Right? So this is the complete equation for the nitrogen fixation. Now the question arises here that this process it is highly energy requiring, और ये जो energy यहाँ पर utilize हो रही है ये कौन provide करेगा? That it will come from the host cell or it will come from the bacterial cell. Yes. So there's energy which is required for the nitrogen fixation. It is provided by the host respiration. So after the nitrogen fixation, this formation of the ammonia, you can say the first stable product of the nitrogen fixation, that is ammonia. So now what is the fate of the ammonia in the plant cell? One thing that we have to study here, and next one I have to discuss some of the important points with respect to the respiration of the rhizobium in the soil and in the plant association. When the rhizobium is in free state, free living state, that is in soil, तो free living state के अंदर rhizobium is aerobic. Yes, so it shows the aerobic mode of respiration, and when the rhizobium shows aerobic mode of respiration, under these conditions, nitrogenase is non-functional. तो इन कंडीशंस के अंदर जो नाइट्रोजनेज एंजाइम है दैट इज नॉन फंक्शनल, बट व्हेन द राइजोबियम इज शोइंग द एसोसिएशन विद लेग्यूमिनस प्लांट्स, तो जब लेग्यूमिनस प्लांट्स के साथ एसोसिएटेड है, तो just now I had discussed, now it will show anaerobic mode of respiration. And now under the anaerobic mode of the respiration, nitrogenase enzyme, it is functional. Right? So, कब rhizobium aerobic mode show करेगा और कब anaerobic mode show करेगा? This is very much clear to all of you. Now let's see what is the fate of the ammonia which is formed in the nitrogen fixation. Now at the physiological pH, ammonia, it is easily protonated and results in the formation of ammonium ion. It is converted into the ammonium ions, and these ammonium ions, they are very much toxic. तो ये जैसे अमोनियम आयंस बनेंगे, इन अमोनियम आयंस का यूटिलाइजेशन होगा. Yes, because they cannot be accumulated, they are really very toxic. So these ammonium ions, they are immediately utilized by the plant. One, it is utilized by the plant for the formation of amino acids. And the second one, these ammonium ions, they are used for the formation of amides. So now let's see how the amino acid amide formation, it was performed by the plant cell. So let's see now how the amino acid and amides, they are formed in the leguminous plants. So now amino acid formation. In the amino acid formation, I will be discussing two methods here. One is reductive amination, the another one is transamination. Reductive amination and transamination. Now first of all, let's start with the reductive amination. In the reductive amination, keto acid reacts with the ammonium ions. Keto acids, they react with the ammonium ions in the presence of reducing power, that is NADH or NADPH. And there is requirement of dehydrogenase enzyme. For the dehydrogenase enzyme, I will be writing DH here. Now to explain this amino acid formation, I am citing one example here, and the example which I am citing here, that is for the 5-carbon keto acid. Yes, First example is for the 5-carbon keto acid, that is alpha ketoglutaric acid. Yes, so I am forming the structure of alpha ketoglutaric acid. This is a 5-carbon keto acid. It reacts with the ammonium ions in the presence of this reducing power and the enzyme dehydrogenase. So now what will happen? From the keto acid, the oxygen is removed. Oxygen is removed as water. राइट ऑक्सीजन रिमूव होगा और यहां पर इस पोजीशन के ऊपर देयर इज एडिशन ऑफ द हाइड्रोजन एंड अमाइनो ग्रुप व्हिच रिजल्ट्स इन द फॉर्मेशन ऑफ अमाइनो एसिड. So there is removal of the oxygen and addition of the hydrogen and amino group, which results in the formation of this 5-carbon acid. This 5-carbon acid which is formed from this alpha ketoglutaric acid, this is glutamic acid. तो ग्लूटामिक एसिड की फॉर्मेशन कैसे होगी? दिस इज वेरी मच क्लियर टू ऑल ऑफ यू. लेट्स साइट वन मोर एग्जांपल टू एक्सप्लेन दिस प्रोसेस ऑफ अमाइनो एसिड फॉर्मेशन. नाउ आई विल बी साइटिंग द एग्जांपल ऑफ द 4-carbon keto acid, that is oxaloacetic acid. So this is the structure for oxaloacetic acid. This is a 4-carbon keto acid. 4-carbon keto acid reacts with the ammonium ions in the presence of the dehydrogenase enzyme and reducing power. Again, the oxygen is removed in the form of the water, and at this position there is addition of hydrogen and amino group. हाइड्रोजन एंड अमाइनो ग्रुप दैट इज रिमूव्ड, और ये जो यहां पर अमाइनो एसिड फॉर्म हुआ, दिस एसिड इज एस्पार्टिक एसिड. So this point, it should be very much clear to all of you, because the same thing I am going to repeat in the photosynthesis as well as respiration. So these two reactions which I am discussing here, I will be also discussing in the photosynthesis and respiration. तो अगर 5-carbon keto acid use करेंगे, the amino acid which is formed, that is glutamic acid, and if the 4-carbon keto acid is utilized, the amino acid which is formed, this is aspartic acid. So here the amino acid which is formed, that is glutamic acid. So you can simply say the enzyme which is involved, that is glutamate dehydrogenase, and in this case it is aspartate dehydrogenase. अब ये जो दो अमाइनो एसिड्स हैं, इससे दो इंपॉर्टेंट एमाइड्स फॉर्म होते हैं. सो हियर अलोंग विद दिस आई विल बी डीलिंग विद द एमाइड फॉर्मेशन. सो इन दो अमाइनो एसिड से दो इंपॉर्टेंट एमाइड्स फॉर्म होते हैं. सो नाउ लेट्स सी हाउ द एमाइड फॉर्मेशन कैन अकर फ्रॉम दीज टू अमाइनो एसिड्स. So now let's see how the amide formation is there. Now from the glutamic acid, the amide which is formed, that is glutamine. Now how the glutamine formation will occur? Now this amino acid, it will react with the ammonium ions in the presence of ATP, there is requirement of ATP, और ये जो हाइड्रोक्सिल ग्रुप है, this hydroxyl group is replaced by the amino group which is provided by the ammonium ions. So now instead of the hydroxyl group, in the amide there is presence of amino group. Yes, so we can say that in the amide there is presence of two amino groups, or you can say the amides, they are double aminated keto acid. Yes, see here, ये keto acid है, एक बार amino group add हुआ तो क्या बन गया? अमाइनो एसिड. एक और अमाइनो ग्रुप ऐड हुआ तो क्या बन गया? अमाइड. So you can simply say that the amides, they are double aminated amino acids. They are double aminated keto acid, and the enzyme which is involved here for the formation of this glutamine, this is glutamine synthetase. Now next one, now from the aspartic acid, in the same way there is formation of amide by the addition of amino group, and the amide which is formed, that is asparagine. Asparagine is formed, and the enzyme which is involved here, that is asparagine synthetase. Right? Now glutamine and asparagine, they are the two important amides which are formed in the plant, and you can simply see that ये जो glutamine और asparagine के अंदर जो nitrogen content है वो इन amino acid से ज़्यादा है. Yes, so you can simply say that in the amides the nitrogen by carbon ratio, this is high as compared to the amino acids. Yes, so you see in this case, इस केस के अंदर how many carbons are there? 5, and nitrogen is 1. So it means in the amino acid, nitrogen by carbon ratio is 1/5. But in the case of the glutamine, now there is presence of 2 nitrogen. So in the amide, nitrogen by carbon ratio, that is 2/5. अब जो fixed nitrogen है, yes, the nitrogen which is fixed, अब इसका transport कैसे होगा? Now the fixed nitrogen, mainly it is transported in the form of amides. Because in the amides, the nitrogen content is more as compared to the amino acid. So you please note this down here. Transport of fixed nitrogen, it will occur, 1, through the xylem vessels. Xylem vessels के through होगा, and this transport, it will occur in the form of amides. Yes, amides, this is in the case of temperate legumes. Temperate legumes में ये condition है, like pea is there, clover is there, but in the case of the tropical legumes, in addition to this, uridase की form के अंदर nitrogen का transport होगा, and in the tropical legumes you can write certain example here. One is soybean, another one is groundnut, peanut, and kidney bean. So this was about the reductive amination. कैसे अमाइनो एसिड बनेगा और इस अमाइनो एसिड से कैसे 2 इम्पोर्टेंट अमाइड्स बनेंगे, और जो फिक्स्ड नाइट्रोजन है दैट इज ट्रांसपोर्टेड थ्रू जाइलम वेसल्स, एंड It can be through the amides in the temperate legumes, and in the addition to this tropical legumes के अंदर it is in the form of the urides. और ये जो दोनों compounds का मैंने यहाँ पर नाम लिखा है, they have high nitrogen by carbon ratio as compared to the amino acids. Now I have to deal with the second method of the amino acid formation, that is transamination. So now the second method of amino acid formation. Transamination. Now in the transamination, now keto acid, it reacts with the amino acid. So, or I can say amino acid, it reacts with the कीटो एसिड. तो पहले केस के अंदर हमने क्या पढ़ा? कीटो एसिड रिएक्ट्स विद द अमोनियम आयंस, बट नाउ इन द सेकंड मेथड ट्रांसएमिनेशन, अमाइनो एसिड रिएक्ट्स विद द कीटो एसिड. Now what will happen here? From the amino acid, the amino group is transferred to keto acid. तो amino group keto acid के पास ट्रांसफर हो जाएगा, राइट? तो जब कीटो एसिड के पास अमाइनो ग्रुप ट्रांसफर हो जाएगा तो कीटो एसिड किस में कन्वर्ट हो जाएगा? आफ्टर एक्सेप्टिंग द कीटो एसिड, after accepted the amino group, now the keto acid is converted into the amino acid, and the keto group of keto acid that is accepted by the amino acid, so it means now the amino acid, it is converted into the keto acid. You are getting this point? जो amino acid है वो change हो जाएगा keto acid के अंदर, and the keto acid, it is converted into the amino acid. So I will try to explain this by writing one reaction here. So I will try to explain this reaction in little bit of more detail. Let's say this is amino acid. Amino acid will give amino group. So this is amino donor. Now this amino donor, it will react with the keto acid. Yes. This is keto acid. You know that a keto acid will now accept amino group. So you can also call it as amino acceptor. So यहां से जो अमाइनो ग्रुप है वो ट्रांसफर हो जाएगा कीटो एसिड के पास, और कीटो एसिड के अंदर जो कीटो ग्रुप है वो ट्रांसफर हो जाएगा अमाइनो एसिड के पास. So it means amino acid will accept the keto group, so it will be changed into this. It is changed into keto acid. और जो कीटो एसिड है वो अमाइनो ग्रुप एक्सेप्ट करेगा. After accepting the amino group, now the keto acid is converted into the amino acid. Right? And the enzyme which is involved in this reaction, the enzyme which is catalyzed this reaction, that is transaminase enzyme. तो रिडक्टिव एमिनेशन के अंदर कौन सा एंजाइम है? डिहाइड्रोजिनेज. एमाइड फॉर्मेशन के अंदर सिंथेटेज, एंड फॉर द ट्रांसएमिनेशन द एंजाइम व्हिच इज इन्वॉल्व्ड, दैट इज ट्रांसएमिनेज एंजाइम. नाउ इन मोस्ट ऑफ द केसेस जो अमाइनो एसिड अमाइनो डोनर का काम करेगा, दैट अमाइनो एसिड इज ग्लूटामिक एसिड. Yes, the glutamic acid is major amino donor in the transamination reactions. Now here I can ask one question. If you have glutamic acid, right, and it reacts with the oxaloacetic acid. And the transamination reaction will proceed. Then what are the products they are formed? Now glutamic acid will give the amino group to oxaloacetic acid, right? तो oxaloacetic acid जब amino group accept कर लेगा, so it is converted into aspartic acid, right? Glutamic acid amino group दे देगा oxaloacetic acid को, but the keto group, it is transferred to this compound. So after accepting the keto group, now it is converted into the keto acid. It is very well known to you that the 5-carbon keto acid will be formed, that is alpha ketoglutaric acid. So this was all about the amino acid and amide formation. Now here I will be discussing some of the important points with respect to the ammonification. So now the next one, ammonification. Now plants और animals के अंदर nitrogen present है in the organic form. Now when the plants and animals they will die, that organic form of the nitrogen is added in the soil. So after the death of plants and animals, organic form of nitrogen, it is added in the soil. So mainly soil के अंदर जो नाइट्रोजन प्रेजेंट होगा ऑर्गेनिक फॉर्म में प्रेजेंट होगा, दैट इज प्रोटीन्स, राइट? तो यहाँ पर अब हम देखेंगे कि ये जो प्रोटीन्स है, जो ऑर्गेनिक फॉर्म ऑफ नाइट्रोजन है, ये सॉइल के अंदर कैसे कन्वर्ट होगा इन ऑर्गेनिक फॉर्म के अंदर, दैट इज अमोनिया थ्रू द एक्टिविटी ऑफ माइक्रो ऑर्गेनिजम्स. Yes, organic form of nitrogen, it is converted into the inorganic form, that is ammonia, through the activity of microorganism, and this process is known as ammonification. Now this ammonification consists of 2 steps. Now the first one, that is proteolysis. Proteolysis, as the name suggests, there will be breakdown of protein. It involves protein breakdown. So protein का breakdown होगा with the help of the enzymes, that is protease. Proteases, these protease enzymes, they are secreted by the bacteria, and this formation of peptides. Now the peptides, they will further undergo breakdown with the help of the enzyme, that is peptidase. Again, bacterial origin enzyme, and these peptidases, now they will convert the peptides into the subunit, that is amino acids. Now in the amino acid, now let's see how this nitrogen is released. Now these amino acid will undergo second step. That second step is deamination. Now in the deamination, now the amino group is removed from amino acid. So let's see, this is amino acid. Yes, this is amino acid. यहाँ से amino group remove हो जाएगा. It is released in the form of ammonia. और जब amino group remove होगा, instead of the amino group, now there is addition of oxygen here. So now this amino acid is converted into keto acid after the addition of oxygen. Yes. So this process of the formation of the ammonia from the protein, this is known as ammonification. Let's write the name of 4 bacteria which are involved in this. One is Bacillus ramosus, then bacillus vulgaris, another one that is bacillus mycoides, right? So अब ये जो अमोनिया सॉइल के अंदर फॉर्म हुआ है, लेट्स सी व्हाट इज द फेट ऑफ दिस अमोनिया. अमोनिफिकेशन के थ्रू अब अमोनिया फॉर्म हो गया, बट अमोनिया इज नॉट द मोस्ट प्रेफर्ड फॉर्म ऑफ द नाइट्रोजन विच कैन बी एब्सॉर्ब्ड बाय द प्लांट. Yes, तो ये जो ammonia है, now it is converted into the preferred form of the nitrogen, and that preferred form of the nitrogen, that is nitrate. So ammonia, some of it will enter the atmosphere, volatilize, atmosphere में enter करेगा, and some of the ammonia, or you can say the major part of the ammonia, it is converted into the nitrate, and the process which is involved in this, that is nitrification. Yes, so the conversion of the ammonia to the nitrate occurring in the soil, this is known as nitrification. Ammonia is converted into the nitrite first, and this chemical reaction, it is performed by the bacteria like Nitrosomonas and Nitrococcus. Now this nitrite, further it is converted into the nitrate. That this step, it is carried out by Nitrobacter and Nitrocystis. So this oxidation reaction which is performed by these bacteria, yes, through this oxidation reaction there is release of energy. There is release of energy, and this energy is utilized by these bacteria for the carbon dioxide fixation. तो इट मीन्स ये जो बैक्टीरिया है, दे कैन फॉर्म देयर फूड थ्रू द एनर्जी विच इज obtained फ्रॉम केमिकल रिएक्शन. So this category of bacteria, very well known to all of you, they are known as chemoautotrophs. For the synthesis of food, they will use chemical energy. Now, along with the bacteria, at this step, the fungi can also carry out the conversion. So, at the first step as well as first step, the fungi which can work, that is Aspergillus flavus. Means nitrification, it can be performed both by the prokaryotic as well as eukaryotic microorganisms. अब ये जो नाइट्रेट सॉइल के अंदर फॉर्म हुआ है ना, दिस नाइट्रेट इट इज द मोस्ट प्रेफर्ड फॉर्म ऑफ द नाइट्रोजन विच कैन बी एब्सॉर्ब्ड बाय द प्लांट. सो एक इस नाइट्रेट का फेट क्या होगा, प्लांट विल एब्सॉर्ब दिस नाइट्रेट, and second is that some of the nitrate, it is converted into the gaseous nitrogen. Some of the nitrate, it is converted into the gaseous nitrogen. It is lost from the soil, becomes the part of the atmosphere, and the process is known as denitrification. And here in this process, the microorganisms which are involved, one is Thiobacillus denitrificans, and the another one, that is Pseudomonas denitrificans. So let's see, जब nitrate plant absorb करेगा, so what will be the fate of the nitrate in the plant body? So nitrate अब plant ने absorb कर लिया. Now what is the fate of this nitrogen in the plant? Plant will utilize this nitrogen for the formation of amino acids. The amino acids will in turn form proteins. But for the formation of amino acids, there is requirement of ammonia. There is requirement of ammonium ions. So it means जो प्रोसेस अभी सॉइल के अंदर हुआ, अमोनिया से क्या बनाया? नाइट्रेट. अब इससे रिवर्स प्रोसेस होगा प्लांट के अंदर फ्रॉम द नाइट्रेट. नाउ अगेन देयर विल बी फॉर्मेशन ऑफ अमोनिया एंड दिस प्रोसेस, this fate of nitrogen in the plant, this is known as nitrate assimilation. Yes? So, in the plant, now the nitrate, it is converted into nitrite. Nitrite, it is converted into ammonia. Ammonia, it will be changed into the ammonium ions, which can be utilized for the formation of amino acids. So there should not be any confusion here. Protein से ammonia form हुआ soil के अंदर, that is ammonification, and that was performed by the microorganisms. Then ammonia से nitrate form हुआ soil के अंदर, that was again performed by the microorganism. पर एक बार जब नाइट्रेट प्लांट ने एब्सॉर्ब कर लिया, अब प्लांट ने इस नाइट्रोजन को यूज करना है फॉर द फॉर्मेशन ऑफ अमाइनो एसिड्स. पर अमाइनो एसिड्स बनाने के लिए दोबारा क्या करना पड़ेगा? अमोनिया फॉर्म करेगा. एंड व्हाट दिस प्रोसेस इज नोन एज? दिस इज नाइट्रेट एसिमिलेशन. But you have to keep this in mind. This nitrate assimilation, it will occur in the plant body, right? तो ammonification, nitrification कहाँ हुआ? Soil के अंदर. And the nitrate assimilation, it will occur in the plant body. Let's see what are the enzymes which are involved at these steps. Nitrate is converted into the nitrite with the help of the enzyme that is nitrate reductase. Reduction will occur. There will be addition of hydrogen, there is addition of electrons, and at this step there is requirement of enzyme that is nitrite reductase. Both nitrate reductase and nitrite reductase, they are metalloflavoprotein. Both of them, they are metalloflavoproteins, right? Now in the nitrate reductase, the metal which is present, that is molybdenum, and here iron and copper. Nitrate reductase, it is present in the cytoplasm. The location is in cytoplasm, but nitrite reductase, this is a chloroplast based enzyme. So it means we can simply say this part of the nitrogen metabolism, it will occur in the green leaves. Yes, this part of the nitrogen metabolism, it will occur in the green parts of the plant. Now this process requires hydrogen and electrons. This is reduction, and in the case of the first step, the electrons and hydrogen, they are provided by NADH2, and here electrons, they are provided by ferredoxin. This is an electron carrier which is associated with the photosystem 1 in the plants. तो ये photosystem 1 के साथ associated है. Out of these two enzymes, this nitrate reductase, it is an inducible enzyme. Inducible enzymes means ये एंजाइम प्लांट के अंदर तभी प्रोड्यूस होगा जब प्लांट के अंदर प्रेजेंट होगा सबस्ट्रेट फॉर दिस एंजाइम. एंड व्हाट इज द सबस्ट्रेट हियर? दैट इज nitrate. Means when the plant will absorb nitrate substrate, then only there will be formation of this enzyme. This is known as inducible. But nitrite reductase, whether the substrate is present or it is absent, it is always present in the cell. It is always formed in the cell. So this is a inducible enzyme. So this is all about nitrogen metabolism. Now here I would like to ask you one question, that which is the most preferred form of nitrogen which is absorbed by the plant? तो सबसे ज़्यादा preferred form of nitrogen which can be absorbed by the plant, that is nitrate, right? And if the question is which is the biologically most usable form by the plant, then what you will say? अब सबसे ज्यादा जो फॉर्म यूज्ड हो रही है दैट इज अमोनिया. यस, क्योंकि अमोनिया से ही आगे अमोनियम आइंस बनेंगे और बाद में क्या बनेंगे? अमाइनो एसिड. सो द प्रेफर्ड फॉर्म फॉर एब्जॉर्प्शन दैट इज नाइट्रेट एंड द मोस्ट प्रेफर्ड or the usable form that is ammonia. So this is all about the chapter mineral nutrition. In the next class, I will be starting with the new chapter that is photosynthesis in higher plants. Thank you so much.